Well, hey there, it's John here, and I'm back in the Eastman Discovery Lab at the Hands-On Discovery Center with another activity that you guys can try right at home. Today is actually Pick Strawberries Day, so of course the star of our episode today is this bright, delicious, gorgeous strawberry. We are actually going to be using this to extract the DNA material from the cells inside of this strawberry. DNA, or deoxyribonucleic acid, is kind of like the code that makes living organisms grow up to what they are. So this strawberry, it's coded through its DNA to grow up to be a strawberry, unlike me, who was coded to become a human. We can actually extract the DNA from inside of the cells using just household ingredients. We can create what is called a lysis buffer, or literally a mixture that will break apart the cells using our favorite chemical, water. Here I have about 90 milliliters of water, and we're going to top it off to about 100 milliliters with Dawn dish detergent. This is what's going to help dissolve the cell walls of the cell and actually break open the nucleus where the DNA is stored. And the last ingredient for our lysis buffer is just a little bit of salt. Here I have it's a healthy pinch. I would say it's about 1/4 of a teaspoon. And we're just going to gently mix that together. This is going to be the chemical part of extracting the DNA, but probably the most fun part is the physical extraction. Grab yourself a Ziploc sandwich bag and your strawberry, and you can chop the little green part off if you want, but it doesn't really matter. Drop it into the sandwich bag, and we are going to physically break down the strawberry. How do you do that? Smush it! Just knead it and pinch it and roll it around. Don't go too crazy because you don't want to burst your bag because then you'll have strawberry guts everywhere. But you want to make sure that all the big clumps are kind of broken down, turn it into kind of like a strawberry jelly, really. And after you've kneaded it and mixed it and pinched it for about 5 minutes, you can go ahead and add your lysis buffer right into the bag and keep kneading for a couple extra minutes. There we go, and you might need help from a friend because sometimes the sandwich bags get a little tipsy and they'll fall all over. But just continue to mush and squish until you have a thoroughly crushed up mixture. Then we're going to filter it. You can grab a funnel and maybe a coffee filter and line the funnel with that so it'll catch all the big clumps. We're going to go ahead and pour that in and get it filtering out, but it does take a little while. You don't want to rush this step because you don't want your filter to break either. But luckily, I've already gone ahead and filtered a sample here. Here we have mostly just the liquidy part, broken down those cell structures, and we've used the lysis buffer to break open the nucleus. The last thing that you're going to need is some isopropyl alcohol, and you want this cold. I literally just took this out of the freezer, and it is so cold. You're going to gently pour a little bit of this isopropyl alcohol. Notice I'm pouring it down the side so it doesn't just totally mix into the strawberry solution. And give it a couple seconds. If you look really close, you'll actually notice there's kind of this light band, this kind of lighter colored, that's the isopropyl alcohol, floating on top. And after a couple minutes, some of the isopropyl alcohol will start to turn kind of this milky white color. That's the actual DNA of the strawberry precipitating out, rising out of the strawberry solution. And after a few seconds, you can kind of use maybe like a popsicle stick. Here I have a little pipette. You can kind of twirl it around, and you might notice some goopy strands. That is the DNA, again, precipitating out of the strawberry mixture. Using my pipette here, I can actually scoop up some of this, and I have a little test tube that I can actually deposit that DNA material in, and I can keep it for as long as I want. But I've literally taken the DNA from these strawberries just using stuff that I have at home. And you can try this with strawberries. They work super well because they actually have multiple copies of their DNA in the cell. But you can try it on blueberries, or bananas work very well too. So try this one out at home, guys. Let us know how it works for you. Hit that thumbs up button if you like the video, and subscribe to our channel so you never miss a new video update. And check the description below where we're going to have all kinds of science standards related to DNA extraction. Also down in the description we'll have some information on how you guys can help us keep the science alive. But I'm about to go pick some more strawberries to extract their DNA. Bye, guys! It's just a giant booger.
That's exactly what it looks like when you pick it
Oh,
up.
gross.
That's DNA!